Okay, so hello everyone. Today in this video, we're going to discuss about human immunodeficiency virus, which is HIV. So before going through HIV, let me tell you that HIV is also known as lentivirus, and the HIV basically what HIV disease is AIDS. Okay, this is known as also known as lentivirus. Okay, and this HIV human immunodeficiency virus basically belongs from subgroup of retroviruses. Belongs from subgroup of retroviruses. Now you might be thinking, what is this retroviruses? Retroviruses is basically a reverse transcribing virus. This is basically reverse transcribing virus. Reverse transcribing virus. This means, for suppose when we are basically converting, like we have transcription, that is DNA to RNA is basically what? DNA to RNA is transcription. But this is basically reverse of this. Now here we are forming DNA from RNA, as here we are forming DNA. So RNA we are forming from DNA, but here in the reverse transcribing virus we have what? We are forming DNA from RNA. So from RNA the DNA is formed. So that's why here one enzyme is used, that is reverse transcribing virus. Okay, or here enzyme is reverse transcriptase. Enzyme is what? Reverse transcriptase. Right here. Reverse transcriptase enzyme. Okay, so now here the immunodeficiency virus HIV basically contain, means the HIV structure basically contain following parts. So if I can go with the first part, so which is envelope. First part is envelope. So now let's discuss the first part in more details. Then we will go with the second part. The envelope is basically what? Envelope of HIV is lipid bilayer. Lipid bilayer, and HIV do not contain their own lipid bilayer, means HIV do not contain own double membrane, but it is the host derived. Host derived. Membrane is what? Membrane is host derived. Matlab it, we can say it got enter into one cell and it get the membrane of the cell. So this is basically what? This is the host derived. Okay, so now if I can go with what, yep, these envelope basically contain the spikes. Okay, this envelope contain the spikes. Spikes are basically one type of, we can, spikes are basically the protein, glycoprotein, we can say. Those are present, or one is embedded in the double membrane layer and second is outside of double membrane layer. So if I can talk about which type of spikes we have. Okay, so here we have two type of spikes. One is glycoprotein 41, another is glycoprotein 120. So these are basically two type of glycoprotein or spikes which are present in the membrane. So now let's discuss about these. So yep, spikes basically contain two type of protein, as I told you, GP120 and GP41. So now GP140 or 120 is basically, it has the work like it, GP120 act like the head receptor. It is basically act as head receptor, and this GP120 we can say bind to the host cell. This because it contains the receptor, so this bind to the host cell. Bind to host cell. Okay, and so this, when this is bind to the host cell, so GP120 is also known as docking protein. Remember that this is question which can be asked. GP120 is also known as docking protein. Fine, I can show you with the help of diagram. First let me tell you what is GP GP41. Okay, so GP41 is basically transmembrane protein. This is transmembrane protein. Transmembrane protein, which basically help in the fusion of host cell and the GP120. Fusion of host cell, होस्ट सेल मेम्ब्रेन जो है उसकी फ्यूजन करवाता है किससे HIV के साथ. फ्यूजन ऑफ होस्ट सेल मेम्ब्रेन विद HIV. So this help in the fusion. Okay, so now here I'm going to share one diagram with you so you can go with it. So yeah, let me share here the diagram. Okay, so here you guys can have a look. I have shared the diagram with you. So this is GP120, which is basically GP120, which is at the upper side of the protein, glycoprotein, and GP41, which is embedded in the membrane, which is transmembrane. Okay, this help in the fusion, and GP120 help the it can contain the receptor, so it can easily bind to the host cell. Okay, so now let's discuss about basically yeah, these both spikes are basically right here. These both spikes are basically encoded by virus. These both spikes are encoded by HIV virus. These are not host derived. Remember this, mostly people think these are also host derived, as HIV membrane is also host derived, so these are host derived. No, these basically contain spikes, and these are encoded by virus, and these are not derived, not host derived. Okay, these are not host derived. So now let's move towards basically the structure as I told you. So this basically contain yep, the capsid of this. Here you can see the capsid. Yep, this is basically what this is the capsid. So it contain basically 9 gene. Capsid contain 9 gene, and from those 9 gene basically here we have what from those 9 gene 3 gene are basically these help in to form the structure to form the structure. However, remaining 5 gene help in their application. Remaining 4. Remaining 6 gene help in replication. Okay. So now here if I can give you bit concept of we can say HIV bind with what. So here you can go with it later on, but yep, here if I have what here if I have one cell which is T helper cell. And this is what this is the HIV, we can say one spike of HIV, and now here we have receptors. These receptors will bind inside this, and this we can say GP41 will help to the fuse this inside the cell. So the fusion of this is done by GP41, and these are combined bind by GP120. Okay, so yeah, because now here you can see these T helper cells are what T helper cell also contain the receptor which is CD4, which are cluster differentiation 4. Okay, so now here I hope this is clear to you guys. Now we should go with second part which is capsid. So let's discuss second part which is capsid. Okay, so yeah, this basically capsid is basically made up of what? This capsid is made up of protein, and this capsid have basically contain 9 gene as I told you. So if I can write here, capsid is basically proteinous code. This capsid is basically proteinase coat and which is made up of what? Which is made up of P24, like which is made up of not glycoprotein but simple protein 24, made up of P24. Okay, and now it is basically it includes the two identical strand of RNA. Like HIV, we can say virus contain two identical strand of RNA virus. So this includes the two strand of RNA viruses. So this is enclosed work in the protection or enclose the genome of HIV. And if I can go with, so yeah, these two we can say identical strand of RNA are not complicated or not opposite. Okay, so yeah, if I can go with it, yeah, this capsid also contain one matrix. So in the HIV virus, we have another thing which is matrix. So here matrix is basically the surrounding layer of capsid. Now if genome is we can say enclosed by the capsid, so capsid is also enclosed by the matrix. So this is the surrounding layer of capsid, which is made up of what? Now this matrix is made up of protein 17. Made up of protein 17. Now this matrix, what is the work of this matrix? So matrix basically play crucial role in what? In stabilizing the capsid. Capsid को एक stable करके रखता है, stabilize करके रखता है. Stabilizing the capsid. Okay, so if I can show you this capsid or matrix in this diagram, so here you can see we have our GP424, which is capsid, and this capsid is surrounded by one matrix which is GP117, which is matrix. Fine. So I hope this is also clear to you guys. Now we should go with what? Now we should go with genome. Okay, so now let me tell you the third part of this, that is genome. Okay, so HIV basically possesses two identical strands of RNA, which are basically known as genome. Two identical RNA strand. Okay, and due to presence of two RNA molecule, HIV is also known as diploid. As we know, okay, diploid is basically what? Diploid is something which contain two things, like two in numbers. So yeah, HIV basically contain two RNA strands, so this is also known as diploid. Diploid, okay. And now HIV possesses two strand RNA. Now which type of this RNA? So this is basically positive sense RNA. This is basically what? This is positive sense RNA. Okay, so now here this is all about genome. Now if I can go with what? If I can go with enzymes, which enzymes are present here? So now let's see the enzymes. Fifth, we have enzymes. Okay. So yeah, these enzymes are basically HIV contain three enzymes which help for the replication. HIV contain their own. HIV के पास अपने तीन enzymes होते हैं जो कि को help करवाते हैं किसके अंदर? Which help in replication. Three enzymes help in replication. Okay, so if I can go with you, so yep, these are basically number one is reverse transcriptase, which is RT virus. Oh, sorry, RT enzyme. Reverse transcriptase. Okay, another we have now, this is basically the enzyme. What is the work of this enzyme? So this enzyme basically work in what? This basically help the enzyme HIV to convert RNA into DNA. To convert RNA into DNA. Okay, convert into DNA now, which is basically cDNA. Now cDNA is basically what? Complementary DNA. Complementary DNA. RNA convert this into complementary DNA. Now RNA sequence will name the complementary DNA of, we can say complementary strand of DNA. So now let me tell you about what. Now let me tell you about integrase. Second enzyme we have integrase. So yeah, this enzyme basically help in what? This enzyme we can say help when cDNA is formed from RNA. cDNA is formed. Okay, so it converts the cDNA into double-stranded DNA. Now cDNA is basically complementary DNA. This means cDNA is one strand. From RNA, the cDNA is formed, which is one strand only DNA. So DNA work on the two strand, double strand. So yeah, this integrase help to convert cDNA into double strand DNA. Okay, double strand DNA, and this is integrated into what? This is integrated into host chromosome by the help of enzyme, which is integrase. Integrated into host chromosome. This is integrated into host chromosome by the enzyme, we can say enzyme, which is known as integrase. Okay, now here you can see, now third enzyme we have, what third enzyme here? We have basically protease. Third enzyme is protease. Now this is basically the enzyme which help in the replication. Now this cleavage basically large protein molecule into smaller, means just cut or we can say break the large protein molecules into smaller protein subunits, smaller protein molecule or subunit. Now the enzyme used to cut these large smaller protein, that is protease enzyme. So I hope that it is clear to you guys. Now let me tell you one more thing here. So yeah, remember that HIV virus, we can say, do not contain proofreading system. HIV virus do not contain proofreading system. Means in our human body we have proofreading system, that is DNA polymerase. DNA polymerase. If any mutation occurs, so DNA polymerase help in proofreading, but HIV do not contain proofreading DNA polymerase, so that's why the mutation occur time to time. So then the antigen basically variations occur because it do not contain the proofreading system. No proofreading system. No proofreading system. So I hope till that it is clear to you guys everything. So in the next video we're gonna discuss about the life cycle of HIV. Till that, bye bye.